Hello, everybody. My name is Angel Perez, and today I'm going to be presenting a presentation on boosting flavonoids and terpenes in plants and how we can use the ultraviolet spectrum in fruits and vegetables to stimulate secondary metabolites. A little bit about me is I grew up poor but inherited a huge love for plants. I'm graduating with a bachelor's in biology with a minor in psychology. And I hope to obtain a master's program at either Utah State University in space biology or crop science, and also, if willing and able, if I get accepted at Pennsylvania State University for plant pathology. Either or seem really great opportunities, but I'm currently working at a pharmacy in South Jordan and at the Division of Wildlife from May to September doing plant identification. As you can see here, the top right hand picture, some of these plants have been alive since the year 2000. They're in my mom's garden, and we moved back to California back in that year when I was about 5 years old, and we planted some of these from around that time period, and they're still alive today. This was that summer, and then here's me at the division. Cavemen couldn't time period, and they're still alive today. This was that summer, and then here's me at the Division of Wildlife. Moving on, though, believe it or not, that cavemen couldn't grow with UV, but you can. So the reason why I put this here is that we haven't had any recent nominal breakthroughs in agriculture. We had in the Stone Age from 12,000 to 3000 BC, the first agriculture plant revolution, where rice paddies, potatoes were grown in the Andes. And then the second agricultural revolution, we had, the seed drill being instilled into plots. Then we had, in the third agricultural revolution, this was driven with a lot of GMOs, a lot of different types of light spectrums. But what we failed to do here is we were growing for yield and efficiencies, not necessarily for health benefits. With that, heading into the fourth agricultural revolution, which we're currently undergoing, we're instilling farm automation, but not really a push as to the nutritional benefits. So, though cavemen couldn't grow with UV or save 15% on car insurance, what you're going to come to learn through my presentation is that you can grow with UV, and you can grow even more greater efficiencies than 15% for your secondary metabolites, starting with flavonoids. There are over 6,000 flavonoids in the kingdom Plantae. They're broken down into 6 categories, and they're associated with taste and color, but specifically they aid in UV protection and have some pretty good medicinal benefits, as you can see here. I've listed the locations of where flavonoids can be found, such as on the orange, you got a lot of carotenoids that build on the outer skin, and flavonoids called the flavito right here. And then we have a lot of the essential oils and oil glands that build up through secretory cells here inside in that white dermal layer. Those are where a lot of buildup of oils such as terpenes are composed, and we'll learn about those. But let's look at the 6 different categories and some of the food sources that you could actually find flavonols, flavones, flavonones, anthocyanins, and isoflavonones. There's a lot of health benefits which we can dive into, but they would be too long to go into a list of. I've actually clipped a lot out of my presentation, so we'll go ahead and cover some of the other ones, just some specific ones. So focusing on some of flavonoids' medicinal properties and how they work is they bind like ligands. They help fight dependency on drugs because they're natural botanical precursors that bind, as seen in the picture, a ligand to a receptor that have pain relieving effects, mood stabilization effects. Sometimes they're used in diabetic medications, specifically in green tea, and that works as an insulin imitator. But one of the most highly abundant ones, like camphorol and quercetin, they form antioxidant and anti-inflammatory capabilities. They're found in broccolis and beans, and essentially you can find a lot of literature about how they prevent long-term degenerative diseases. They lower the risk of long-term heart disease. But one of the favorite ones that I love to cover is it actually works to modulate pro-inflammatory cytokines. It actually decreases the release of cytokines, so that way you don't get as much inflammation and a lot of those that are related to cancer over time. But they're not the only actual chemical we'll be focusing on today. We also have the terpenes, which are volatile aromatic compounds associated with aromas. They're increased pathogen resistance in all plants, and these are actually ancient conserved pathways in all animals and plants, the terpene production pathways. They're called the MEP and MEV, which we're going to learn about. But they also serve as UV protection, such as flavonoids. They both have medicinal properties and they both have UV protection. Two of the ones that I wanted to cover today were philandrine. I thought it was kind of funny because it has a eucalyptus-like aroma that smells like koala bears. That's this one right here. And then we had caryophyllene, clove-like aroma found in clove flavor. And it's also found in pepper and cannabis. And then some of the other terpenes that you can find, there was monoterpenes, sequitur terpenes, but the ones that you can find in humans are like retinols, like vitamin A. So there's terpenes found everywhere, but they're actually the most abundant molecule ever measured by mass on Earth. Now, after I had studied them, I've learned that algae were actually the first ones to produce terpenes as nature's sunblock protection, because in shallow pools, when there was less ozone, UV caused mutations that allowed terpene production to take place so that algae could continue to live. But some other ones here were arlamoline. That's one's found on orange peels. It gives you that citrus-like smell, and it's used in household products. So these are everywhere you look. Now, the importance of terpenes is that they contain medicinal properties. They can serve immunomodulatory roles. They reduce the risk of cancer, and they actually serve as great pain relievers as well because they inhibit descending pain pathways. Here, I've provided a picture of citrol that acts on these CB1 and CB2 receptors. CB1 is for neurotransmission, so it's used as a stress and anxiety reliever sometimes in some papers. And then specifically where they've actually found that it binds like a ligand is to the CBR2 receptor. This is used as anti-inflammatory and immunosuppressive roles. And as you can see here, citrol and this one off to the right, beta caryophyllene, those both have been documented to bind to CB2 and have highly antidepressive like effects, and it actually has very high anti-inflammatory properties. But there's just a list of other receptors that it can work on. One of the most recent papers I learned about was the SARS-CoV beta caryophyllene trials that they're doing that actually inhibit or decrease long-term risk factors for those that have suffered through COVID. The problem, though, is we don't have a very big focus on secondary metabolites in agriculture. We grow for the bulk. During that third agricultural revolution in the 50s, we found that visible light is more efficient for sugars and carbohydrates due to the Starks-Einstein law, where we found that, if one photon can excite one electron here in this picture, to drive it into the photosystem pathway. In this case, if we use 4 photons to drive 4 electrons, first we accomplish the first step and it makes ATP within the visible light spectrum as a product. But it still needs to undergo one more jump into the photosystem 2 pathway, and that requires either highly excited far red photons or just visible light spectrum photons. Another 4 of them would propel this electron here up into the second stage photosystem. That was known as the Emerson enhancement effect, one of the most recent light revolutionary experiments that they ran, that far red photons are more efficient for growing yields of products. As you can see here, I provided summary of spectral effects. It enhances cell expansion. but it doesn't necessarily have a huge impact on secondary metabolites. So while photosynthesis is primarily associated with glucose and starch production, it also produces secondary metabolites as byproducts. UV radiation has been seen to be way more effective at producing secondary metabolites. Furthermore, they even found that one UV photon is equivalent to about 100 visible light photons, so it doesn't even have to be used in such intensity. So since there's such a high value in secondary metabolites, but such a focus on growth production for our fruits and vegetables, I propose that we should use the UV light-induced approach to grow fruits and vegetables. To understand the UV spectrum's benefits, a brief explanation is needed though on how visible light spectrum affects photoreceptors. So what are photoreceptors? They're light-sensing proteins that are found in practically all plants, and they synthesize secondary metabolites in response to specific wavelengths. They bend and respond to them. There's UV-specific photoreceptors that get affected, and those ones that we'll cover are going to be listed right here, UVR8 and Crypticum cries. As you can see here, the UV spectrum runs down here from 200 to about 400, and they're classified as UVB rays, UVA rays in this color format. And photoreceptors have been seen to improve fruit quality, the actual biomass of crops food security, and they've even increased pesticide resistance, because if you can stress out a plant enough, it starts to do some very funky defensive mechanisms, decreasing the reliance on pesticides. So by activating these UV-specific receptors, we can actually produce more secondary metabolites. So the UV-specific photoreceptors that I'd like to cover that we've gone over in that previous little photograph is UVR8 and cries, which are stimulated by UV, as you can see here. They actually create a precursor molecule called Hi5 that is the first committed step to any flavonoid production. As a result, if you stimulate these two photoreceptors, you get more flavonoid production. And this was seen in a study, and a little depiction here on this picture I have for you guys, where on a sunny day with high UVA rays, you get increased chemical compounds such as terpenes and flavonoids and antioxidants. But on a cloudy day, you get more photosynthesis happening and a breakdown, causing more sugars to be produced. That is known as photoinhibition. And when photoinhibition happens, terpene production is actually driven up and photosynthesis is driven down. But when photosynthesis is up, terpene production and second metabolites is driven down. So while we have UV-specific photoreceptors that drive flavonoid production, we also have enzymes that aid in terpene production as a focus. So just a little recap on what enzymes are is they synthesize secondary metabolites from precursors. As you can see here, a substrate binding to an enzyme into the enzyme substrate complex, releasing a product. But they do sometimes require some form of activation, and in this case, we have UV-specific enzymes that are activated by different wavelengths of the UV spectrum. Now, recalling that the terpene production pathway is a conserved pathway, I chose the MEP and MEV paths, which have UV-specific enzymes, and I'm going to explain to you guys how we drive up terpene production using those specific enzymes. As you can see here, we have DXS, IPPI, GPPS, and FPPS, which are all enzymes upregulated by UV. In the MEP pathway, we can get the product such as monoterpenes by those three enzymes, DXS, IPPI, and GPPS, such as limonene, that smells like lemon, and pinene, that was found in our pine trees. And then we have the MEV pathway, which creates rare sequitur terpenes through FPPS, and such and some of those are known as humulene. We had beta caryophyllene that we covered. And by stimulating these enzymes, this results in increased mono and secretory terpene concentrations in all plants, since these are conserved pathways. So while using enzymes and photoreceptors is a practical approach to increasing secondary metabolites, we got to understand the discrepancies in the quality of UV light that we're using, because there are UVA, UVB, and UVC rays, as seen here. We've got UVC, UVB, and UVA. And the results depend on the duration that we use UV for, as well as the wavelength that is being used, and the situation, if it's being instilled during their growth phase, pre or post harvest. So some evidence of using the UVA spectrum from 315 nanometers to 400 nanometers was an experiment by Chen that measured secondary metabolite development in lettuce at 365 nanometers after 13 days. And believe it or not, in all spectrums, UVA 10 and durations, UVA 20 and 30, there was increased metabolite production, with UVA 20 being the highest here with almost 29% increased production. But as you can see, the results are dependent on the duration that they used it for, whether 10, 20, or 30 minutes. But they did find that UVA is more beneficial than just visible light. Here you have UVA 20 compared to the control, but any UVA spectrum, as well as their weights, their leaf area, their overall development increased using some supplementary UVA spectrum, as you can see here in the charts. And specifically, the total flavonoids and phenolic content was our interest, and you can see 1.62 compared to the 2.05 over here, and so on. It's almost a 25% difference. We can, however, also use the UVB spectrum, and this was seen in an experiment done by Castagna in 2013, and he studied an experiment on tomatoes using UVB for 1 hour per day for 22 days. And tomatoes were harvested at the green stage, and they found that using UV treatments actually increased carotenoids, which are a type of flavonoids in the peel. As you can see here, using UV, you can drive carotenoid production. So they found that UVB is a practical spectrum for increasing secondary metabolites, even after it's been pulled off the vine, not only during development. And the reason why is because red light works on different photoreceptors that drive chlorophyll production, as seen to the far left, where UV light drives secondary metabolites such as carotenoid and pigmentation production, like antioxidants. Lastly, I'll be covering UVC because it's gotten such a big misnomer for being used in the post-COVID world as a sanitizer. So it can also be used as a secondary metabolite producer. And the reason why they discovered this was Kim et al. Back in 2008 harvested some tomatoes and used UVC on them. And they actually found that not only does it improve production of actual secondary metabolites, but it delayed the degradation of vitamin C after 7 days by 30%. So it can prolong the second metabolites that are produced throughout the lifespan of the fruit and vegetable, and not just used as a sterilizer within the agriculture industry for killing E. coli and salmonella, and COVID in this case, because they used it for eradicating a lot of the viruses in medical environments. So to wrap it all up, in conclusion, UV treatments on UV-specific photoreceptors such as UVRA and CRISE can increase flavonoids, while UV treatments on UV-specific enzymes, DXS, IPPI, GPPS, and FPPS increase terpenes. And we also got to be careful with what spectrum we're using, UVA, B, and C. Though they all yield far more secondary metabolites than the visible light spectrum, they all yield different results. So we got to be careful with that. As well as the UV light-induced approach is an effective way overall to grow fruits and vegetables to increase secondary metabolites for our health benefits. And then the future outlook that we're looking on, if we do use the UV light induced approach, we can build less reliance on pesticides with increased fruit pathogen resistance by having higher terpenes on their skins, which are highly anti-pesticide. And we can build more equitable cost per fruit with nutrient density. No more will you have to pay for big flavorless fruit. And doing so, we'll build flavors the likes of which we have yet to see. And here are some other advantages and drawbacks that are currently holding us back from using the UV light induced approach. Thank you for watching my presentation, by the way.